To the world of biology through Jaguar classes from Jagran Public School, Noida. Today, in this video lecture, which is the second part of molecular basis of inheritance, in this chapter, today we will cover the topic packaging of the DNA helix. In earlier part 1 of this chapter, we have discussed about the structure of polynucleotide chain, Watson and Crick model of DNA helix, and chemical composition of DNA with Chargaff's rule. After today's class, in which we will talk about the packaging of DNA helix inside the cell, prokaryotic as well as eukaryotic, the learning outcomes we are expecting after today's class are you will be able to identify the components of DNA and as well as you will be able to understand the arrangement of the different components in the macromolecule, and you will also able to gain the knowledge of the physical structure and dimensions of DNA with its packaging. You will be able to know the difference between heterochromatin and euchromatin, and the students, uh, you should have the knowledge of inheritance and variation that we have already learned in the previous chapter 5, principles of inheritance, that will be used. This is the basis of for this chapter also. As in earlier chapter you have studied that how in different patterns the traits are inherited from one generation to another generation, but what is the mystery behind this? Whatever factors Mandel has discussed or thought about responsible for this pattern of inheritance, that particular factors are actually the biomolecule DNA. Means what are the names of the two nucleic acid found in living organisms, that also you are already knowing, RNA and DNA, and which molecule is responsible for the transmission of characters from one generation to the next, that we are talking about in this chapter. Now let's start with our today's class to know about how the DNA is our today's class with second part of molecular basis of inheritance. In first part of this chapter, we have already studied about the structure of DNA and Watson and Crick model of DNA structure. Now in this part, we will focus on how DNA packaged inside a cell. As you know that DNA is such a long polymer. It is one interesting fact that how this 2 meter, approximately 2 meter thread in our cell is packaged. But the length of this DNA is not in all the cells is 2 meter. Its packaging is very interesting. Now let's start to know that how this molecule is packaged inside a cell. In prokaryotes, DNA held with proteins, nucleoid, large loops held by proteins. Now you can see here in prokaryotes, this is just like a single DNA in nucleoid region and which is packaged inside a capsule. But in eukaryotes, it is found in highly supercoiled form, and in this prokaryotic cell it is just like a loop. In prokaryotic, if we will see the packaging of DNA like in a bacterial cell, you can see that how a single loop like structure as a single chromosome present inside the nucleoid region, which is not surrounded by any nuclear membrane because it is a primitive type of cell, as you have already studied in earlier classes. Now come to know how it is packed inside the eukaryotic. In eukaryotic cell, there are 200 base pairs of DNA helix which are repeatedly organizing themselves in the form of a unit known as chromatin, which are seen in the form of beaded like structure like beads on a string that you can see here how these beads like structure are present inside the nucleus of the eukaryotic cell. The chromatin, which is a thin thread like form of the DNA, is highly condensed or supercoiled form in the form of chromosome structure at the time of division. This particular condensed form in the form of chromosome is seen. Here you can see that nucleosome, DNA helix, histones which are packed together in the form of that chromatin, and these histones proteins are present along with the DNA inside the nucleus. The typical structure or 3D structure of that DNA packed DNA inside the cell is known as nucleosome. This is a nucleosome unit you can see here in the form of a core of beta histone molecule as well as uh, H1 histones and that wrapped DNA along them is present, which is forming one nucleosome. And what is an octamer, which is actually a positively charged basic proteins called histones present inside a octamer, which form a unit of 8 molecules, due to which it is known as histone octamer. DNA wrapped around histone octamer in this form that you can see here in this diagram. And in this, the question is, what is an octamer? You can see here an octamer unit of the packaged DNA inside a eukaryotic cell is packed in this way. Here you can see that H2B, H2A, H3, H4, these 8 nucleosomal structures are present and linked with that DNA which is wrapped around it. This is one beaded structure with one octamer, and this is the another beaded structure with the another octamer. Here only DNA is without histone protein is present here, and this particular part is known as nucleosome, which forms a bead like structure, or this particular unit of the packaging of DNA inside a eukaryotic cell is known as 8 histone molecule plus 146 nucleotide base pairs of the DNA. Means around that uh, octamer unit 146 to 200 base pairs are wrapped here in one octamer unit. And on the basis of this, you can see the structure of the DNA inside a cell having two different stain. One portion is lightly stained and the another portion is darkly stained. Where this octamer beaded like structure is present, the part of the DNA at that particular place is darkly stained and it is known as heterochromatin. while this loosely packed structure, where no histone proteins are arranged or packed with the DNA strand, that particular portion of the packed DNA inside a eukaryotic cell is considered as euchromatin. We can compare between these two structures, euchromatin and heterochromatin, inside a cell. These two regions can be compared as euchromatin is loosely packed portion of the DNA, while heterochromatin is tightly packed portion of the DNA. Here, the euchromatin is transcriptionally active, while heterochromatin is transcriptionally inactive state of the DNA. It stains light, which were euchromatin stains light while heterochromatin stains dark. These are the three important points of comparison between euchromatin and heterochromatin when we are studying about their packaging inside the cell. And euchromatin versus heterochromatin, you can see here in this structure the orange colored regions indicate the heterochromatin, which is the we can say that tightly packed structure and which is stained uh, more darker as compared to the euchromatin region. And euchromatin region is shown here by the purple color strand here, and in between that there is a centromere. In this way, you can see that the packaging of DNA is going on inside a eukaryotic and prokaryotic cell. And this is really a mysterious process inside a cell where such a bigger molecule or such a big polymer is packed in such a supercoiled or condensed form, which we cannot see through our naked eyes. To see or to observe this, we need some special aids or instruments like compound microscope and electron microscope to study its detailed structure. And this is the mystery of the nature of this molecule, biomolecule, which is the main basis for the inheritance. That what is the difference between chromosome, gene,
Chromosomes,
and
DNA, and genes. Chromosomes are thread-like structures present in the nucleus of both animal and plant cells. They are made of protein and a single molecule of deoxyribonucleic acid, typically referred to as DNA, the genetic material of the organism. During metaphase, a chromosome consists of two components known as chromatids, which are in every way identical. Chromosomes will be visible under a microscope only when the cell is dividing. Cells containing a nucleus, which are eukaryotic cells, have linear chromosomes. Prokaryotic cells, those without a defined nucleus, have circular chromosomes. Each chromosome has a constriction point known as the centromere, dividing it into two sections or arms. The shorter arm is labeled the P arm and the longer is labeled the Q arm. During mitosis and meiosis, the chromosomes will duplicate and appear as a four-arm structure. Somatic cells contain diploid chromosomes, meaning they have double the number of chromosomes. Gametes, often referred to as sex cells, contain haploid chromosomes, meaning half the number. DNA is a macromolecule polymer made of subunits known as nucleotides. These nucleotides are made of three units: a phosphate group, deoxyribose, and nitrogenous base. The nucleotides are arranged into chains or strands. Each strand has a backbone made of alternating groups of deoxyribose sugar and phosphate groups. The sugar molecule of one nucleotide is linked with the phosphate of the next nucleotide. There are four nitrogenous bases in DNA: adenine, guanine, referred to as purines, cytosine, and thymine, known as pyrimidines. The bases are attached to the sugar molecule. Two strands of nucleotides will pair together in a specific way. Adenine pairs with thymine and guanine pairs with cytosine. AT and GC base pairs are said to be complementary. These paired DNA strands will form a double helix, which looks like a ladder that has been grabbed at each end and twisted in opposite directions. A stretch of DNA or DNA sequence contains instructions for making a protein and is known as a gene. Genes carry biological information that is copied and transmitted to the next generation each time a cell divides to form two daughter cells. Summary: chromosomes are thread-like structures that are present in the nucleus of both animal and plant cells. The paired DNA strands form a double helix, which looks like a twisted ladder. The instructions to make a protein on a stretch of DNA or DNA sequence are known as a gene.
With that, you will be able to understood about the DNA, chromosome and gene.